Hello, my name is Sungjin Lee, and thanks for inviting me to this wonderful conference. It is my great pleasure and honor to share the recent work that has been conducted in our lab. So in this talk today, I will discuss about one of our research topic regarding a novel neuronal circuit controlling the sleep physiology in mouse models using optogenetics. So before I explain the essential findings in our study, I would like to briefly cover some of the basic concepts that will be dealt with in this presentation. First of all, in sleep research, sleep stages are mainly classified into either non-rapid eye movement or non-REM sleep and rapid eye movement sleep or REM sleep. Non-REM sleep is usually characterized by little or no eye movements, dominance of parasympathetic nervous system, and it is also well known as deep sleep, when you have really a good quality sleep. And in humans, this non-REM sleep can be classified into 3 stages, where in stage 1 is when people start falling asleep with theta waves becoming the most prominent waveforms in the EEG. And in stage 2, sleep spindles starts to appear, which are 7 to 15 hertz sharp wave ripples formed from the interaction between the cortex and the thalamus. And then in stage 3, delta waves, which are the most lowest brain waves, become most prominent. And this stage is also known as slow wave sleep. On the other hand, REM sleep is characterized by very rapid random movement of the eyes, as well as low muscle tone or muscle atonia, where your muscles get temporarily paralyzed. And this is when people experience very vivid dreams. And in addition, the EEG signals usually show low voltage waves that are very similar to the awake state. Next, I would also like to briefly explain about one of the most common methods that is currently being utilized actively in modern neuroscience, which is optogenetics. Optogenetics, which literally means utilization of gene expression of opsins, which are light-sensitive proteins that are activated by light delivery, was initially developed by a neuropsychiatrist, Dr. Carl Deisseroth at Stanford University. Since its first development, it became one of the most innovative techniques in controlling and manipulating the activity of neurons in animal models, especially in rodents. The basic principle of optogenetics is by virally expressing an opsin encoding gene at a certain brain region that targets certain types of neurons, and we can specifically stimulate or inhibit the activity of those neuron types simply by light delivery at certain wavelengths. Now, based on these concepts, I would like to introduce some of the detailed backgrounds of our research. In our study, we focused on the brain region called the basal forebrain, which is a collective region of heterogeneous nuclei situated in the rostroventral forebrains. This figure depicts the major nuclei of the basal forebrain, or BF region in mice, and most of these structures are also conserved in other mammalian brains, including humans. Various cell types are also involved in the BF, including the cholinergic and the glutamatergic and GABAergic neurons. The cholinergic neurons are most well known due to the degeneration of BF cholinergic neurons, such as in Alzheimer's disease, as shown in previous studies. And the basal forebrain is also known for diverse brain functions, such as cortical activation, attention, motivational salience, memory, cognition, and as well as sleep-wake cycle. So in one of the previous studies, which conducted the juxtacellular recordings of GABAergic neuronal subpopulation in the BF during sleep and wakefulness, it was identified that these BF GABAergic neurons fire at around 40 hertz during wake states and REM sleep. Also, this subpopulation was also active during non-REM sleep as well as at 10 hertz. Another study also discovered that the majority of these BF GABAergic neurons express the calcium binding protein called parvalbumin, or also known as PV. And these BF PV GABAergic neurons project to the cortex. When the actual function of these BF PV neurons were tested, optogenetic stimulation of BF PV neurons at the gamma frequency range 40 hertz induced the same frequency response at 40 hertz in the cortex by synchronizing the activity of the cortical PV interneurons. Moreover, this optogenetic stimulation of BF-PV neurons at 40 hertz significantly increased wake states and decreased the non-REM sleep in mice. However, as mentioned earlier, the function of their firing pattern around 10 hertz during non-REM sleep has not yet been investigated. Other studies have also shown that BF GABAergic neurons project to the TRN, which is thalamic reticular nucleus. The TRN is well known for the generation of theta range frequency around 7 to 15 Hertz, which is the typical frequency for the EEG sleep spindles. This is also based on an earlier study that demonstrated the function of the TRN promoting cortical spindles or delta waves by optogenetics. So based on these studies, we hypothesized that the BFPV neuronal projections toward the TRN might regulate sleep and wakefulness by projection-specific optogenetic stimulation, as shown in this figure. To test our hypothesis, we conducted two types of experiments. For the first type, we gave stimulation at 8 Hertz and 40 Hertz during the light and dark period, respectively, in the basal forebrain. And for the second type, we gave the stimulation specifically on the TRN projecting BFPV neurons at 8 Hertz with 6-hour recording in each 4 time points, each representing the light period, early, middle, and late dark periods. I would like to note that each period during the dark period represents low, intermediate, and high sleep pressure intervals. For selective stimulation of PV expressing neurons, we used the Cre-dependent opsin expression system. Here, we used PV-Cre mice, which express Cre recombinase specifically in PV positive cells. The virus we injected encodes double OXP gene, making it possible for selective expression of the opsin, channeled opsin 2, only in the cells expressing Cre recombinase. As I explained earlier, opsins are basically light-sensitive proteins that can also involve ion channels that respond to specific wavelengths of light delivery. For the EEG recordings and optogenetic stimulation in mouse brains, we implanted the EEG electrodes and the optical glass fiber, then fixed with the dental cement, and then fixed the EEG-EMG head mounts on top of these electrodes that can be connected to the preamplifier for EEG recording. The EMG wires are also inserted into the thoracic muscles in mice. After the virus injection and the EEG head mount implantation surgery, we conducted in vivo EMG EEG recordings with optogenetic stimulation using these specialized recording device and specialized software, and then manually scored their vigilance status classified into wake states, non-REM, and REM sleep stages. Based on the previous studies, we then applied 8 Hertz and 40 Hertz stimulation by using this stimulation protocol. The 8 Hertz stimulation protocol was to mimic the natural induction of sleep spindles, and the 40 Hertz protocol was to induce gamma frequency EEG waves, as shown in the previous slides. After the EEG recording, in order to validate the expression of viral vectors and the neuronal activation by optogenetic stimulation, we performed the immunohistochemistry as shown in these procedures. So after anesthetizing the mouse, we perfused the heart to perfuse the brain blood vessels with PBS. And then after defreezing the brain, we sectioned the brain into thin slices, and then also incubated with the antibodies and mounted on the slides for the microscopic imaging. So with immunohistochemistry, we confirmed that the channel rhodopsin 2 was selectively expressed in the PB positive neurons. Also, the optical fiber tips, which are approximately situated on the BF regions for the stimulation. And co-localization of channel rhodopsin 2 with PV revealed that the channel rhodopsin 2 was selectively expressed in these PV neurons. We also confirmed that the BFPV neurons project to both the cortex and the TRN, as shown in the immunohistochemistry imaging through fluorescence microscopy, as shown here. So this is the cortex region, and the TRN region is shown here below. And to selectively stimulate the BFPV neurons toward the TRN, the PV-Cre mice were injected bilaterally with AAV5 encoding channel rhodopsin 2 in the BF regions with identical volumes. Now, while the fiber optic cannula were bilaterally positioned to TRN to stimulate axon terminals of BFPV neurons, with the immunohistochemistry, here again, we confirmed that the TRN neurons were successfully activated by optogenetics, as shown by the CFOS expression, and these neurons were also receiving direct direct projections from the channel Dawson 2 expressing BFPV neurons. And the implanted optical fiber tips approximately targeted the TRN regions as shown in this figure. Furthermore, we next confirmed that these TRN projecting BFPV neuronal fibers were specifically targeting the PV expressing neurons in the TRN as shown in these figures. So these neurons were successfully activated by optogenetic stimulation of PV neurons as represented by the CFOS expression in the TRN. I'm not sure if you can see clearly here, but if you magnify the screen, you'll be able to see the merged image that shows purple colors and the yellow colors in panel G and H that shows the co-localization of parvalbumin and CFOS, as well as the channel Dawson 2, which represents the inputs from the basal forebrain axonal fibers. We also verified that the bilateral stimulation is more effective than unilateral stimulation for optogenetics. So in fact, the previous member in our lab conducted preliminary experiments with unilateral stimulations, and she showed a slight increase of sleep using this method. So we hypothesized that the bilateral stimulation would induce more powerful effects than unilateral stimulation. And as you can see here in panel C and D, the fold changes of bilateral stimulation were much stronger than ipsilateral and contralateral stimulation in both frontal and parietal regions. Graph E and H are representative time frequency spectrogram induced by the 40 Hz and 8 Hz stimulation respectively. So when we use this optogenetic stimulation protocol, the intermittent stimulation at 8 Hz during the light period induced no changes in any sleep or wake states. However, this stimulation increased non-REM sleep duration during the dark period when the mice are mostly awake and active. Additionally, in contrast to 8 Hz stimulation, 40 Hz stimulation did not induce any significant changes in non-REM sleep, as shown here. Non-REM duration, bout count, bout length were not changed in both light and dark periods. We also investigated the effect of stimulation on sleep spindle density. However, we couldn't observe any distinct sleep promoting or wake promoting effects, which is supposed to be demonstrated by the sleep spindle increase or decrease. Based on these results, we suppose that there may be dual projections of BF to cortex and the TRN. We also performed 6 hours of EEG and EMG recordings with 2-hour optogenetic stimulation at 8 Hz. This stimulation was performed in 4 time periods, each consisting Zeitgeber time 6 to 8, 12 to 14, 18 to 20, and 22 to 24, which are light period, early, middle, and late dark periods on separate days. And then we analyzed their response by 8 Hz stimulation. From these data, we compared evoked spindle range frequencies. And generally, as you can see, our data indicate that the cortical EEG response at spindle range frequencies were higher in the dark period than the light period. So when we analyzed the overall results on non-REM and REM sleep duration with this stimulation protocol, the early dark period, indicated in blue, shows some decreasing trends of non-REM duration immediately after the stimulation period, which implies the improvement of the sleep quality by the stimulation. In the middle of the dark period, indicated in green non-REM sleep, decreased during the stimulation. And in the late dark period, indicated in yellow, showed some non-REM bout count increase, but no significant increase of total duration of non-REM sleep was shown. On the other hand, REM sleep data reflects the effects of non-REM increase and decrease respectively as shown in this bout count results. Additionally, there were almost no difference during the light period in both REM and non-REM sleep. But when we look at our data in a much wider level, which is total sleep duration, only minimal effects were shown during the late dark period as shown here with the increase of sleep bout lengths. Due to these contradictory results from different time points during non-REM and REM sleep by intermittent 8 Hz optogenetic stimulation, I further analyzed other sleep parameters as well. And those indicators were slow wave activity and slow wave energy. Panel A and B show slow wave activity increase during the mid-dark period, whereas panel C and D show slow wave energy increase during the late dark period. Interestingly, during the middle of dark period, although there was no change in slow wave energy, there were some increase of sleep spindle densities as shown in panel E here. So we could say that the non-REM decrease was not due to sleep deprivation. Rather, it was due to the improvement of the sleep quality by increased slow wave activity and slow wave energy. In other words, because of the enhanced sleep quality, the mouse did not require long sleep duration. So finally, as a summary, this figure recapitulates our findings. When we specifically optogenetically stimulated the BFPV neurons only, there were no changes in the light period. But during the dark period, it slightly induced non-REM duration, also the non-REM bout counts, and there were no changes in the sleep spindle. However, when we specifically stimulated optogenetically the TRN projecting BFPV neurons, although there was no change in the light period, we could observe the slow wave activity and slow wave energy increase during the early dark period, as well as decrease of non-REM duration in the mid-dark period, and the slow wave activity increase. And also, in the late dark period, the sleep out length also increased. There were no significant difference in the sleep spindles. Now, based on our results, we suppose that the reason why only dark period stimulation was effective rather than light period was because the TRN neurons are hyperpolarized by adenosine, which is a molecular marker for sleep pressure. Since the sleep pressure is high during the dark period due to the wakefulness in mice, during this wake state, adenosine accumulates and causes hyperpolarization of the TRN involved in the thalamocortical network. To further explain our main findings, we can summarize that the 40 Hz optogenetic stimulation with a 5-second train of the TRN projecting BFPV neurons increased total wake duration. However, 8 Hz optogenetic stimulation with a 1.25 second train with 7.5 second intervals in the TRN induced stable sleep spindle rhythms and an increased amount of total non-REM sleep. This was based on the previous studies published earlier. Now, in our finding, intermittent stimulation at 40 Hz in the BFPV induced no change in both wake and sleep. This is because the two stimulation patterns, which is the 40 Hz wake promoting frequency and the 8 Hz sleep promoting frequency pattern, offset each other's effect. So that is why no effect was observed. For further questions regarding our research, we wonder whether are the cortical projecting neurons and the TRN projecting neurons in the BF the same population. So in order to investigate that, we would conduct a retrograde tracing of the AAV mCherry virus injection in the cortex, and that is projected from the BF. And also we can conduct a retrograde tracing from the TRN to investigate the circuits that project from the BF to both the cortex and the TRN in a retrograde manner. Also, we want to further find out whether the delta frequency range stimulation could enhance non-REM sleep in the basal forebrain. And we could also conduct that experiment as well as in the TRN by stimulating the TRN in the delta frequency range. So further study of the subregions of TRN is also required, including the retrograde tracing of the channelrhabdosin 2 virus injection in the TRN, and also just by stimulating the BF. We want to know that whether there are retrograde projections from the TRN to the BF, and as well as the PV neural projection from the BF to TRN. So as a conclusion, we can conclude that driving BFPV neurons at 8 Hz optogenetically and in an intermittent fashion could modulate non-REM sleep, including continuity of non-REM sleep, slow wave activity, and slow wave energy. A novel role of TRN projecting parvalbumin BF neurons in modulating non-REM sleep may be associated with increasing slow wave activity or slow wave energy rather than changing sleep spindles. And altogether, these findings suggest that the TRN projecting PV neuronal subpopulation in the basal forebrain among the BFPV neurons could operate as sleep promoting neurons in addition to their well-known wake promoting function depending on their firing frequency and pattern. So this would be all for my presentation, and I would like to thank my advisor, Tae Kim, who is the PI of our lab, and our previous lab member, Ji-Ae He, who was a master's student who graduated from our lab last year. Actually, she has done most of the work and experiments for this study presented today. Lastly, I would like to say thanks to all of our lab members as well. Thank you for listening, and please feel free to ask any questions. Thank you for your attention.